Hi, we are Team 3, and we're going to talk about the use of compost enhanced with GMOs for the bioremediation of soils polluted by pharmaceutical compounds.
Nature results to be our main source of oxygen, food, among all the essentials for human thrive. But sadly, due to human pressure on nature, their surface has been altered by almost 75%. It has been stated by the Natural History Museum in London that over the last 50 years, nature's capacity to support us has plumbed, starting from the reduction of the air and water quality, the depletion of the soils, and the low levels of pollinators. Due to the aforementioned, it was decided to focus in the 15th Sustainable Development Goal, whose main objective is to restore and promote the sustainable use of terrestrial ecosystem, sustainably manage forests, combat desertification, halt and reverse land degradation, and halt the loss of biodiversity, and in focus especially in the 15.3 goals target, which states that by 2030 we have worked in the restoration of the degraded land and soil, including land affected by desertification, drought, and floods, and strive to achieve a land degradation neutral world. It is for this reason that the presence of different xenobiotic compounds found in the surface and in residual water in the metropolitan zone of Mexico City, Mexico, such as ibuprofen, naproxen, and diclofenac, and the identification of different microorganisms, will be studied in order to adapt a composting technique for the elimination of emerging pollutants and to help with the proof of efficiency in drug reduction.
In order to meet the aforementioned objective, it is imperative to use tools such as microorganisms using recombinant technology. Currently, in the environment, there are several species aimed at the environmental recovery of various species. Hussain et al. Mentions that genetic engineering has the potential to improve bioremediation and gives as an example a species of Pseudomonas to which genes from other 4 wild variants were introduced, which improved from 10 to 100 times its ability to degrade oil, which is a xenobiotic compound. In our case, we will focus on medicines such as ibuprofen. Microorganisms have already been identified in the compost that can degrade the xenobiotic compounds. Marimon describes the species Klebsiella oxytoca and Alkaligenes fecalis subspecies fecalis inoculated in composting have shown good results in the degradation of ibuprofen and azithromycin, respectively. Therefore, it can be deduced that through recombinant technology, we can use microorganisms such as those mentioned above. In this way, soil degradation by xenobiotics will be combated, thus contributing to reach the 15.3 objective of the 2030 Agenda of the United Nations.
Contamination by organic micropollutants is a problem that has been clearly documented in the metropolitan zone of Mexico City. Among these contaminants, xenobiotic compounds such as pharmaceuticals can be found in surface and groundwater, and ibuprofen, naproxen, and diclofenac are the more common pharmaceutically active molecules that are present in wastewater. The presence of these compounds in residual waters are mainly due to improper disposal practices via sink, toilet, or rubbish bins. Since these xenobiotic compounds are found in residual waters and the station of residual waters are not efficiently equipped with processes that can eliminate pharmaceuticals, these are released to the environment, contaminating the soil, and the major accumulation of these molecules can be found in the muds used in agriculture and composting. Several techniques of composting have been related with the degradation of pharmaceutical compounds, often influenced by microorganisms such as bacteria or fungi. Hussain et al. Found that the remediation of organic xenobiotic compounds may be enhanced by the activity of genetically modified organisms, such as plants and microbes, as well as recombinant technology. In this matter, Xiong et al. Reviewed the potential application of microalgae in the removal of pharmaceutical contaminants, engineering this organism in order to express enzymes of the laccase family, which are oxidoreductases that are substrate-specific. Since this is an algal enzyme, it only requires oxygen from the environment as electron acceptor and only produces water as byproduct. Thus, they are considered green catalysts. Moreover, it has been reported that the laccase gene from Mycoleptora thermophila has been transformed into Saccharomyces cerevisiae in order to apply the enzyme in the degradation of polyaromatic hydrocarbons, and this procedure derived on the 8-fold increase of the expression of laccase. Other microorganisms that have been reported to enhance the degradation of pharmaceutical compounds are Klebsiella oxytoca and Alkaligenes fecalis subspecies fecalis.
As previously mentioned, the effectiveness of bioremediation of agricultural soils contaminated with xenobiotics can be improved by applying some compost made from rice straw and sewage sludge. Within this composting was isolated a bacteria Phainobacillus barcinonesis that allowed the obtaining of more stable compost. The inoculation of the strain of Saccharomyces cerevisiae expressing the fusion protein P4 cellulase A of barcinonesis P3BY48741 favor the degradation of organic matter during composting. The two genetically modified strains of Saccharomyces cerevisiae, the P4 lipase, a fusion proteins of Bacillus subtilis, and the P4 cellulase A fusion proteins of P. barsonensis, were eliminated through the process, which is indicated that the composting is a safe technique to study their behavior and thus prevent genetically modified organisms from being transferred to the environment. This result is a more stable compost that absorbs 7.65 milligrams of oxygen per 1 gram of dry mass. Other important results are that no significant differences were found in pH, electrical conductivity, auxiliary organic carbon, and total nitrogen. On the other hand, the effect of oxytocin on the elimination of the ibuprofen that is added to the high concentration during the composting process, this microorganism was isolated and identified by the same mixture with this drug. To compare the degradation rate of the drug through the experiment, a control was prepared with the presence of the inoculum, but to which the drug was also added. The only difference found in the physical, chemical, and biological parameters between the two mixtures were the obtaining of higher humic acid, fulvic acid ratio in the mixture inoculated with the oxytocin compared to the control mixture, as well as a faster start to the thermophilic phase, which started 10 hours earlier in inoculating mixture and maintaining temperatures above 55 degrees. The results obtained in the biomutation test of composting with microorganisms capable of using the drugs as a sources of carbon and nitrogen for growth shows an improvement in the degradation efficiency of azithromycin and ibuprofen during the composting process. Something that was observed in the bulk composting is something necessary is the reinoculation of microorganisms through the process to obtain rates of degradation of the fasted drugs.
The results obtained in this work open up important possibilities to solve the problem of xenobiotics contaminants found in waste through composting, and to obtain improvements in this process through the inoculation of the aforementioned microorganisms. Summarizing this research will be sustained on the aforementioned findings and will be focused on the solution on the contamination of soil by residual waters containing pharmaceuticals such as ibuprofen in the metropolitan zone of Mexico. Using compost enhanced by the presence of genetically modified organisms such as bacteria, microalgae, and yeast. Our objective is to comply with the United Nations 15 SDG, which talks about the conservation of terrestrial ecosystems based on previous studies of soil being contaminated with active compounds in Mexico. It is expected to combat this with bioremediation. Compost enhanced with GMO bacteria, microalgae, and yeast achieved by recombinant technology. Our specific objectives are: create a composting technique for the elimination of xenobiotics in residual waters, use genetically modified Klebsiella oxytocin and Alkaligenes fecalis subspecies fecalis for the degradation of ibuprofen and azithromycin. After the bioremediating procedure, it is expected to restore the contaminated soils. Thank you.